Okay, let's unpack this. Have you ever truly stopped to consider what happens inside your muscles when you lift a weight, sprint across a field, or, even just stand still?
Right.
It's far more intricate than simply muscle
Oh,
contracts.
absolutely. Much more going on.
Today, we're taking a deep dive into the incredible world of skeletal muscle physiology. We're going to pull back the curtain on its microscopic architecture, how it generates immense force, how different muscle types enable everything you do, and even the body's remarkable ability to, well, repair itself after a tough workout.
And what's truly fascinating here is moving beyond just what happens to really understanding why it matters so profoundly.
Exactly.
From the intricate molecular dance of proteins
Yeah.
all the way to the devastating impact of certain genetic conditions. This isn't just about memorizing facts.
No, definitely not.
Our mission really in this deep dive is to explore the provided material with the kind of depth needed to truly master this information.
Right, like the elite professionals in this field.
Precisely. We'll be uncovering every concept, every detail, aiming to ensure you gain a comprehensive, expert-level understanding.
That's the goal. So get ready for some surprising facts, some powerful insights, and probably a whole lot of aha moments as we explore the amazing engineering of your own body right from the ground up. Okay, so to truly master skeletal muscle, we really need to start with the basic blueprint.
Makes sense. Foundations first.
We know muscles are designed to convert chemical energy into mechanical energy, mainly using ATP to generate force.
The universal energy currency, yeah.
And we're focusing specifically on skeletal muscle today, the kind that's under your voluntary control. And interestingly, it looks striated or striped under a microscope. That's because of its internal organization.
Those characteristic stripes, yeah. Very telling.
And these are the muscles that act on your skeleton, like your biceps and triceps working across your elbow joint.
If we zoom out just a bit, look at the bigger picture. The clever way muscles attach to bone is key.
Through tendons, right?
Exactly. Via those tough fibrous bands called tendons. And that's what allows for that huge range of motion we have.
Okay.
We talk about the origin. That's usually the muscle's more stable attachment, closer to the body's midline, the proximal end.
Got it.
And the insertion is the more mobile attachment, further away, more distal.
So like the biceps originates up near the shoulder and inserts down on the forearm bone.
Precisely. So a flexor muscle, like that biceps, it decreases the joint angle.
Bending the elbow.
Right. And an extensor, like the triceps on the back of the arm, does the opposite, increases the angle, straightens it out.
Okay, here's where I think it gets really interesting. The whole hierarchical organization, it's like layers within layers.
It really is, like a rope made of smaller ropes.
Exactly. Imagine your whole muscle like a thick, powerful cable. Inside that cable, you don't just have one solid piece. You've got numerous smaller bundles.
Fascicles.
Right, fascicles. And each one is wrapped in its own protective layer of connective tissue, the paramysium. And that's where the blood vessels and nerves run, too, feeding that specific bundle.
Okay, so then if you look inside one fascicle, you find the individual wires.
The actual muscle cells.
The muscle cells, or muscle fibers.
Yeah.
And they can be incredibly long, right? Like up to 25 centimeters.
Yeah, surprisingly long cells. And each single fiber is then wrapped in its own delicate layer, the endomysium.
Okay, endomysium around the fiber, paramysium around the fascicle.
And then the epimysium encases the entire muscle, the whole cable, as you put it.
And right at the ends, all these connective tissues, epi, peri, endoes, they all merge together.
To form that strong tendon.
Which anchors the muscle to the skeleton. And this structure isn't just for neat packaging, is it?
Not at all. Those connective tissues are rich in things like elastin and collagen. They provide really important passive tension.
Ah, so they help resist stretching.
Exactly. They prevent the muscle from overstretching and contribute to its overall elasticity. It's structural and functional. Okay, so a natural next question then. How is the force actually transmitted? From those tiny wires through the whole cable.
Good question. The myotendinous junction is absolutely key here.
That's where muscle meets tendon.
Precisely. It's a highly specialized region where the muscle fiber membrane, the sarcolemma, folds extensively.
Like little fingers interlocking.
Kind of, yeah. It dramatically increases the surface area for contact.
So it spreads the force out.
Exactly. Distributes the force more effectively, reduces stress concentration. But the striated appearance, those stripes we mentioned,
Yeah.
that comes from the organization inside each muscle fiber, Within each fiber, you find these long bundles called myofibrils,
Okay, bundles within the fiber.
right, running the whole length.
Yeah.
And these myofibrils are divided longitudinally into repeating units,
The famous sarcomeres.
the sarcomeres.
Where?
The fundamental contractile units, they're marked by two dark lines called Z lines or Z discs.
And these are tiny, like 2 micrometers long?
Incredibly small, yeah. Think of millions of these tiny engines lined up end to end within each myofibril.
It really is like a perfectly choreographed micromachine inside every cell.
It absolutely is.
So inside that sarcomere, you've got the 2 main types of protein filaments.
The actin and the myosin.
Right, the thin filaments, mostly actin, extending from those Z lines.
Hmm.
They make up the lighter colored I band. And then smack in the middle, you have the thicker myosin filaments. They form the darker A band.
And the zone where they overlap is critical. That's where the interaction happens for contraction.
Okay. Within the A band, there's that lighter zone in the middle.
The H band, or H zone, yeah. That's where you find only myosin, no actin overlap when the muscle is relaxed.
And that dark line right in the very center of the sarcomere.
There, M-line. It acts like an anchor point, helping to organize and align those thick myosin filaments. Keeps everything tidy.
Tidy and incredibly functional. So surrounding each of these myofibrils, there's another critical network.
Ah, yes, the sarcoplasmic reticulum, or SR. Absolutely vital.
It's like the muscle cell's internal calcium reservoir.
Exactly. It's this intricate intracellular membrane network that meticulously stores and releases calcium ions. It controls the switch, essentially.
And getting the signal to the SR involves those T-tubules.
Right. Transverse tubules, or key tubules, they are invaginations, like little tunnels of the outer muscle membrane, sarcolemma.
So they run deep into the fiber.
Deep inside, yeah. They're essentially in contact with the extracellular space, bringing the electrical signal inwards.
Okay. And the parts of the SR right next to the T-tubules, those are the important bits for calcium release.
Those are the terminal cisternae, or like swollen sacs of the SR packed with calcium.
And there's a tiny gap between the T-tubule and the SR.
Yeah, just about 15 nanometers. Very close association.
And this whole setup T-tubule sandwiched between 2 terminal cisternae that has a name, right?
That's the triad, a very specific and crucial structure. It's the precise site where the electrical signal, the excitation, gets coupled to the calcium release, the contraction trigger.
Excitation-contraction coupling happens right there at the triad.
That's the spot. And the rest of the SR, the longitudinal parts, they're packed with a pump.
Circa.
Circa, exactly. The sarcoplasmic endoplasmic reticulum calcium ATPase. Its job is to pump calcium back into the SR after contraction.
Which is essential for relaxation.
Absolutely crucial. Without Circa, the muscle would stay contracted.
Okay, let's zoom in even further, right down to the filaments themselves, the thin actin filament. It's like a twisted ladder.
Yeah, a double helix. It's formed from many individual globular actin molecules, G-actin, linking up to form fibrous actin, F-actin.
And there's another protein running alongside it, nebulin.
Nebulin, yes. It's a long, thin protein thought to act like a molecular ruler, regulating the precise length of the thin filament.
But the real control system, the on-off switch, involves other proteins on that thin filament.
The regulatory proteins, the tropomyosin and the troponin complex.
Okay, so tropomyosin, these are long, thin molecules.
Very long dimers, yeah. They lie along the groove of the actin helix, and in a resting muscle, they physically cover up the sites on actin where myosin wants to bind.
Blocking the interaction.
Exactly. Each tropomyosin molecule covers about 7 actin binding sites, and they link up head to tail along the whole filament.
And sitting on each tropomyosin is the troponin complex.
Right, the troponin complex. It's a 3-part switch: troponin T, troponin I, and troponin C.
T, I, and C, what do they do?
Troponin T binds the whole complex to tropomyosin. Troponin I is inhibitory. It helps hold tropomyosin in that blocking position.
And troponin C, that's the calcium sensor.
That's the key one. Troponin C has 4 sites where calcium can bind. 2 are high-affinity sites, usually bound to calcium or magnesium. Even at rest, they seem to control the interaction between troponin I and T.
Okay.
The other 2 are lower-affinity sites. These only bind calcium when its concentration really spikes inside the cell, like after that SR release.
And that binding is the trigger.
That's the trigger. Calcium binding to troponin C causes a conformational change in the whole troponin complex.
Which pulls tropomyosin out of the way.
Essentially, yeah. It shifts tropomyosin deeper into the actin groove, exposing those myosin binding sites.
Unlocking the potential for contraction.
Precisely. And there are other helper proteins too, like tropomodulin at one end, CAP-Z protein at the Z-line end, and alpha-actin helping anchor it all at the Z-line. It's a very precisely assembled structure.
Incredible detail. Now, what about the thick myosin filaments? How are they organized?
Also incredibly organized. They aren't just floating freely, they're anchored.
By what?
By titin, an absolutely enormous protein, one of the largest known.
Titin.
Yeah, it connects the ends of the thick myosin filament all the way to the Z-line. It's crucial for maintaining the structural integrity and alignment of the myosin filaments within the sarcomere.
Wow. So it acts like a spring or an elastic band?
It does have elastic properties, yes. Contributes to passive tension and helps pull the sarcomere back to its resting lengths after stretching. It's vital. So vital, in fact, that defects in titin
Lead to diseases.
yes, known as technopathies. They're linked to certain serious muscular dystrophies and cardiomyopathies.
Shows how important that organization is.
Exactly.
And the myosin molecule itself is complex too, right? Heavy chains, light chains.
Very. Each myosin molecule has 2 large heavy chains and 4 smaller light chains.
Okay.
The heavy chains wind together to form that long rod-like tail section, which makes up the backbone of the thick filament.
Right.
But then, at one end, the N-terminal end, they fold into these globular heads that stick outwards.
Those are the bits that interact with actin?
Exactly. Those heads are the business end. They contain the binding site for actin and the site that binds and hydrolyzes ATP, the ATPase activity.
And the light chains.
There are 2 associated with each head.
Yeah.
One is called the essential light chain, which is crucial for stabilizing the head and for that ATPase activity. The other is the regulatory light chain.
Regulatory. What does it regulate?
It can be phosphorylated, meaning a phosphate group can be added to it.
Yeah.
And this phosphorylation can actually influence the kinetics of the myosin-actin interaction, potentially sensitizing it to calcium.
So fine-tuning the contraction.
A way to modulate it, yes.
Okay, so the thick filament itself, how do these myosin molecules assemble?
They associate tail to tail first, in the very center.
Creating that bare zone you mentioned earlier.
Exactly, a central region with no heads. Then they aggregate head to tail, extending outwards from that center.
So you get heads pointing in opposite directions on either side of the M line.
Precisely, a bipolar arrangement. The heads on one side of the sarcomere point towards one Z-line, and the heads on the other side point towards the opposite Z-line.
Ah, and that's critical for pulling the Z lines together.
Absolutely essential. That bipolar design is what allows the myosin heads to effectively pull the actin filaments inwards from both ends, shortening the entire sarcomere during contraction. It's just brilliant biological engineering. Hashtag 2, force transmission, and the critical role of dystrophin. So that raises a really important question. We've talked about the microscopic interactions within the sarcomere.
Generating force at that tiny level.
Right. But how does that force actually get transferred out of the muscle cell to create the large-scale movement we see, like lifting an arm?
Good point. It has to get from the sarcomeres to the tendons somehow.
Exactly. And it happens in 2 main ways. Longitudinally, along the length of the fiber towards the tendon.
Which involves that myotendinous junction we talked about.
Yes, that folded junction handles the longitudinal transmission efficiently. But equally important, maybe even more so for cell integrity, is lateral transmission.
Sideways, to the connective tissue around the fiber.
Precisely. Through structures called costomeres.
Costomeres. Okay, what are they?
They're specialized adhesion sites located along the sides of the muscle fibers, kind of like ribs on the inside of the cell membrane. They effectively link the Z-lines of the outermost myofibrils
So right from the contractile machinery.
directly across the sarcolemma to the extracellular matrix, that network of proteins outside the cell. It involves a complex network of proteins forming these links.
And why is this lateral transmission so important?
Here's where it gets really crucial for your overall muscle health. This lateral force transmission via costomeres is thought to be absolutely vital for stabilizing the sarcolemma itself.
The muscle cell membrane.
Right. It protects it from the mechanical stress, the ripping forces generated during powerful contractions. Think of it as a reinforcing grid.
The protective shield.
A key protective shield, exactly. And defects in these junctions, both the myotendinous junction and these costomeres.
That's where problems arise.
Big problems. Defects in the proteins that make up these structures, including a really critical one called the dystrophin glycoprotein complex, or DGC,
DGC.
are directly associated with various devastating forms of muscular dystrophy.
Ah, so it's about structural failure.
Largely, yes. But these junctions aren't just structural scaffolding, they also contain important signaling molecules. So they likely play roles in cell communication and adaptation too.
Let's focus on that DGC, dystrophin.
Yeah.
You mentioned it's critical.
Extremely significant. Dystrophin itself is a huge protein, about 427 kilodaltons, though it's actually present at pretty low levels, maybe 0.025% of total skeletal muscle protein.
But clearly important despite the low amount. Where is it located?
It's found right on the inner surface of the sarcolemma, cytoplasmic side, and it forms this crucial structural link.
Reaching what to what?
It links the muscle cell's internal actin cytoskeleton
Okay.
through a complex of other proteins in the membrane, the glycoprotein complex part, ultimately connecting to proteins in the extracellular matrix outside the cell, like laminin.
So it bridges the inside to the outside.
Exactly. It acts as that primary stabilizer for the sarcolemma, that shield we talked about, preventing injury or rupture during contraction.
Makes sense.
And beyond just structure, it might also act as a scaffold, organizing other proteins involved in cell signaling pathways. It even binds to neuronal nitric oxide synthase, or NNOS.
Okay. So its absence or defect has major consequences. You mentioned Duchenne's.
Duchenne's muscular dystrophy, DMD, is probably the most well-known example. It's an X-linked recessive genetic disease.
You meaning it primarily affects boys?
Primarily affects boys, yes. And it's caused directly by a defect in the gene that codes for dystrophin.
So they don't make functional dystrophin protein.
Or make very little, or a non-functional version. This leads to a severe deficiency, not just in skeletal muscle, but also in the brain, retina, and smooth muscle, where dystrophin is normally found.
And the clinical picture is grim.
Very much so. Patients typically show muscle weakness early on, often become wheelchair-bound by age 12 or so. Yes. And tragically, many succumb to respiratory failure or cardiac complications in adulthood, often between 30 and 40 years old. It just starkly highlights how vital this single protein is for maintaining basic muscle integrity. Without it, the membrane is fragile and easily damaged by contractions.
Wow. Are all muscular dystrophies related to dystrophin?
No, not all. While dystrophin defects are common, especially in DMD and the milder Becker muscular dystrophy, other mechanisms definitely exist.
Like what?
Well, for example, there's a condition called limb-girdle muscular dystrophy 2B. This isn't due to dystrophin, but rather a defect in the process of sarcolemma repair.
Ah, so the membrane gets damaged, but the cell can't fix it properly.
Exactly. This is often attributed to a loss or mutation of a protein called dysferlin, which seems crucial for patching up membrane tears. This leads to muscle wasting, particularly around the hips and shoulders, the limb girdle.
Okay. What else?
Defects in titin, that giant protein we discussed.
It's tendinopathy.
Yes, tendinopathies can also cause other forms of muscular dystrophy, like limb girdle muscular dystrophy 2J and tibial muscular dystrophy, which affects lower leg muscles.
Right.
Even mutations in enzymes, like the protease calpain 3. Loss of its activity has been linked to certain myofibrillar myopathies, where the internal structure of the muscle fiber breaks down.
So it's not just about the membrane link. Internal components are critical too.
Absolutely. It truly underscores the complex, intricate interplay of a vast number of proteins. They all need to work together correctly for healthy, functional muscle. A defect in almost any part of the system can have serious consequences.
Okay, let's dive into this core process then. How does that electrical signal, the nerve impulse, actually make the muscle contract?
This is excitation-contraction coupling, or ECC, the crucial link.
Right, so it starts with an action potential, that electrical wave.
Traveling along the sarcolemma, the muscle fiber's outer membrane.
And then diving deep into the fiber via those T-tubules we mentioned?
Exactly. It penetrates right into the core of the cell.
And when that signal hits the triad region, bam.
Pretty much. It triggers an incredibly rapid chain reaction. The key result is a sudden explosive burst of calcium ions, Cas plus AMR, released from those terminal cisternae of the SR.
Released into the main body of the muscle cell?
Into the myoplasm, yes. The cytoplasm of the muscle cell.
And that calcium surge is the direct trigger for contraction.
That's the immediate signal that initiates the actin-myosin interaction.
What's amazing is the speed.
Incredible speed and precision. The action potential itself is super brief, less than 5 milliseconds.
Wow.
But that calcium release follows almost instantly, causing intracellular calcium levels to just skyrocket.
And that peak calcium level triggers the contraction.
It promotes the actin-myosin binding, leading to force generation. This increase in calcium usually peaks around 20 milliseconds after the action potential, initiating what we call a muscle twitch.
A single brief contraction. Okay, so here's the million-dollar question. How does that electrical signal in the T-tubule open the calcium gates in the SR? They aren't directly connected electrically.
Right, that's the fascinating part. It involves a very direct, physical protein-to-protein interaction.
Okay, walk us through it.
The T-tubule membrane contains specialized voltage-gated calcium channels. The main one in skeletal muscle is KV1.1,
KV1.1, also known as the...
the dihydropyridine receptor, DHPR. That name comes from the drugs used to first identify it, but functionally it acts as a voltage sensor.
It senses the change in voltage from the action potential.
Exactly. Now, sitting right across that tiny gap in the SR membrane of the terminal cisternae are the calcium release channels.
The gates themselves.
The gates themselves. These are famously known as ryanodine receptors, or RYR, again named after a substance that binds to them. The specific type in skeletal muscle is RYR1. It's a huge protein complex that forms the actual channel for calcium to flow out.
Okay, so you have the voltage sensor, KV1.1, in the T-tubule, and the calcium gate, RYR1, in the SR. How do they talk to each other across that gap?
This is key. In skeletal muscle, it's not primarily about calcium flowing through KV1.1 to trigger RYR1. That happens in heart muscle, but not really here.
So what happens instead?
It's believed to be a direct mechanical link. When the action potential depolarizes the T-tubule, the KV1.1 channel undergoes a subtle but critical conformational change.
It changes shape because of the voltage change.
Exactly. And the shape change is physically transmitted, likely through protein interactions, directly to the RYR1 channel.
Like pulling a plug.
That's a great analogy. The conformational change in KV1.1 mechanically pulls open the RYR1 channel gate.
And that lets the stored calcium flood out of the SR.
That's the crucial step. It's a direct electromechanical coupling. And recent studies suggest it involves a core complex of maybe just 5 essential proteins.
Which ones?
KV1.1 itself, its helper subunit beta-1a, protein called STAC3, RYR1, another protein called junctophyllin. They form the essential machinery for this coupling.
So STAC3 is involved in that physical link.
It seems to be crucial for the interaction between KV1.1 and RYR1, yes. The voltage sensing part is within KV1.1, but specific loops on KV1.1 seem to interact with STAC3 and RYR1 to make that mechanical connection work.
Amazing. Are there other proteins involved in this region?
Oh, yes. Several others play important roles in fine-tuning it. You have junctophyllin, which, as the name suggests, helps to bridge the SR and T-tubule membranes, maintaining that precise junctional structure.
Holding the machinery in the right place.
Exactly. And inside the terminal cisternae, you have calsequestrin.
What does that do?
It's a calcium-binding protein, but with relatively low affinity. It acts like a sponge, binding up large amounts of calcium within the SR.
Which allows the SR to store much more calcium than it otherwise could.
Precisely. It keeps the free calcium concentration inside the SR low, but the total stored amount very high. This creates a steep concentration gradient ready for rapid release when RYR1 opens.
Clever. Any others?
There are proteins like tridin and junctin also in the SR membrane. They bind to both RYR1 and calsequestrin, potentially anchoring calsequestrin right near the release channel to make calcium buffering even more efficient right at the source. And there's another less abundant calcium-binding protein in there too, histidine-rich calcium-binding protein.
So it's a whole coordinated system just to handle the calcium release.
Highly coordinated. And there's even evidence for another way calcium can get into the muscle cell, particularly during long, hard contractions.
Huh. What's that?
It's called store-operated calcium entry, or SOCE.
Store-operated.
Yeah, it means calcium entry from outside the cell is triggered when the SR calcium stores get depleted. It involves a complex called ORIS-DIM1.
And this happens during things like tetany.
It seems particularly relevant then, yes. While it's not essential for the initial ECC spark, studies suggest that blocking SOCE can reduce the maximal force during really high rates of stimulation.
So it might help top up calcium levels during prolonged activity.
That's the idea. Maybe compensating for some calcium leaking out of the cell or being extruded over time, helping maintain that peak force.
Right. And as we've seen with other systems, when things go wrong here...
The clinical consequences can be severe. Genetic mutations in these key players, KV1.1, RYR1, even STAC3, are linked to some serious conditions.
Like what?
Mutations in KV1.1 are associated with hypokalemic periodic paralysis, where people have episodes of muscle weakness, and also involved in myotonic dystrophy type 1.
Okay.
Mutations in either KV1.1 or RYR1 can cause susceptibility to malignant hyperthermia.
Ah, you mentioned that one, the dangerous reaction to anesthetics.
Exactly. It's a life-threatening inherited trait. Certain anesthetics like halothane or the muscle relaxant cyclinocholine trigger this massive, uncontrolled calcium release from the SR via the faulty RYR1 or related proteins.
Leading to rigidity, high temperature.
Severe rigidity, rapid heart rate, hyperventilation, and dangerously high body temperature. It's a medical emergency needing immediate treatment with dantrolene, a drug that specifically blocks that uncontrolled SR calcium release.
Scary stuff. What about other RYR1 issues?
There's central core disease, another rare inherited condition caused by RYR1 defects. It leads to muscle weakness and, strangely, a loss of mitochondria in the central core of muscle fibers. It seems related to mitochondrial calcium overload.
And didn't you mention STAC3 mutations?
Yes, a specific mutation in STAC3 is found in a rare congenital disorder called Native American myopathy. Shows how critical that coupling protein is.
Okay, one more. Brody's disease. That sounds different.
It is. Brody's disease causes painless muscle cramping and, importantly, impaired relaxation after exercise, especially in the cold.
So the muscles stay contracted.
They have trouble relaxing quickly. It's thought to be due to decreased activity of the SERCA1 pump, the pump that puts calcium back into the SR, often due to mutations in the gene for SERCA1, which is mainly in fast-twitch fibers.
So calcium isn't cleared away fast enough.
That seems to be the issue. And then there's myotonia congenita.
Also involves prolonged contractions.
Yes, painless cramping or stiffness after voluntary movement. But this one is caused by mutations in a chloride channel gene, CLCN1.
A chloride channel, how does that cause stiffness?
Reduced chloride flow across the muscle membrane makes the muscle fiber hyperexcitable, prone to firing repetitive action potentials after the initial stimulus. That causes the prolonged contraction.
Ah, okay. And this is the one with the warm-up phenomena.
Exactly. Interestingly, stress and epinephrine can make it worse, but repeating the contraction a few times can actually relieve the stiffness temporarily.
Fascinating range of issues, just from problems with calcium handling or excitability.
It really highlights how finely tuned the whole system is.
So on the flip side then, relaxation, it all comes down to getting that calcium back into the SR.
Primarily, yes. That's the main job of the SERCA pump.
Which is super abundant in the SR membrane.
The most abundant protein there, yeah. It's distributed throughout the longitudinal tubules and non-junctional parts of the terminal cisternae. It diligently pumps calcium ions from the myoplasm back into the SR lumen against their concentration gradient using ATP.
Using energy to tidy up.
Exactly. And there are helper proteins here too, like sarcoleminin, which might enhance SERCA's uptake by buffering calcium near the pump.
And you mentioned some small peptides that regulate SERCA.
Right. Endogenous micropeptides like phospholamban, sarcolipin, and myoregulin, they act like brakes, subtly decreasing SERCA's sensitivity to calcium.
So they fine-tune the relaxation rate.
They seem to modulate its activity. Phosphorylation of phospholamban, especially in slow-twitch muscle where it's more abundant, can relieve this inhibition, speeding up calcium transport similar to its crucial role in the heart. Sarcolipin is also mainly in slow muscle, while myoregulin is found in both types.
Okay, let's connect the dots. We've got the calcium surge from the SR. How does that actually translate into movement, into the filaments sliding?
Right. The calcium is the key, and the thin filament is the control panel.
So calcium floods the myoplasm, binds to troponin C on the thin filament. That binding acts like a switch, causing tropomyosin to shift out of the way.
Exposing the myosin binding sites on the actin filament, moving towards that cleft between the actin strands.
Okay, the sites are now open. What happens next?
The energized myosin head can finally bind to the actin.
Energized meaning it's already sort of cocked and ready.
Exactly. It's already hydrolyzed ATP and is holding onto the ADP and phosphate. It's primed. When it binds to actin, it forms what we call a cross bridge.
The physical link between myosin and actin?
That's the link. And the formation of this cross bridge is what allows tension to be generated.
Now, you mentioned troponin C's binding sites. Some are high affinity, some low. How does that play out?
Those two high affinity sites, which bind calcium or magnesium even at rest, they seem to mainly control how troponin I and troponin T interact, sort of setting the baseline state.
Okay.
Is the two lower affinity sites that are crucial for initiation. They only bind calcium when its concentration really jumps up after SR release. That's the binding that triggers the conformational change and moves tropomyosin.
Got it. And you also hinted that myosin binding might help move tropomyosin even more, a cooperative effect.
Yeah, that's a fascinating idea. While one troponin-tropomyosin complex controls about 7 actin monomers, the strong binding of myosin head to one of those actin sites might actually cause the adjacent tropomyosin molecule to shift as well.
So one myosin binding could potentially open up sites for other myosin heads nearby.
Potentially exposing binding sites on as many as 14 actin molecules in total. It suggests a positive feedback, a cooperative mechanism, making the process more efficient once it starts.
Very cool. Okay, now for the main event. The cross-bridge cycle. This is the engine, right? The ATP-driven process.
This is the absolute core of force generation and movement. It's a cycle, a series of basically 4 key steps where myosin works like a motor.
Okay, step 1. Let's call it state A, resting state.
State A, resting or ready. The myosin head has already partially hydrolyzed ATP, so it's in this high-energy state, holding ADP in inorganic phosphate. Pi. We represent it as M-ADP pi. It's cocked like a spring waiting.
Waiting for the actin site to be exposed.
Exactly. Which brings us to state B binding. Calcium comes along, moves tropomyosin, exposes the site.
And the energized myosin head snaps onto the actin, forms the cross bridge.
Right. M-ADP pi binds to actin, forming AAM-ADP pi.
Okay, state C. This is the power part.
State C, the power stroke. This is where the force happens. The myosin head undergoes a rapid conformational change. It pivots or swivels the ratchet action,
Pulling the actin filament.
pulling the actin filament powerfully towards the center of the sarcomere, towards the M line. And critically, during this pulling motion, the ADP and the inorganic phosphate, pi, are released from the myosin head.
Ah, so the energy release is coupled to the pull and the product release.
Exactly. This transition to the AM state is the force-generating step. The sarcomere shortens.
Okay, powerful stroke done. What's state D? Gotta detach, right?
State D detachment. A new molecule of ATP comes along and binds to the now empty nucleotide binding site on the myosin head, AM.
And that binding makes it let go of the actin.
Yes. ATP binding causes the myosin head to detach from the actin filament. This detachment is absolutely crucial. Without new ATP, it would stay stuck.
Like in rigor mortis?
Precisely. Rigor mortis happens when ATP runs out and the cycle gets stuck in that attached A or M state, state C, without product release.
Okay, so it detaches. Then what?
Then the cycle repeats. After detachment, that newly bound ATP is immediately hydrolyzed back to ADP and pi, which cocks the myosin head, returning it to the energized state A, M-ADP pi.
Ready to bind again if calcium is still around and the actin site is open.
Exactly. The cycle continues, potentially hundreds of times per second for each myosin head, as long as intracellular calcium levels remain high enough to keep tropomyosin shifted.
And relaxation happens when?
When the circa pumps lower the calcium level. Calcium dissociates from troponin C. The troponin-tropomyosin complex shifts back to its blocking position. Myosin can no longer bind, and the muscle relaxes.
How far does one power stroke actually move the actin?
It's estimated that a single cross-bridge power stroke moves the thin filament about 10 nanometers. Tiny distance, but billions of them working together create macroscopic movement.
Incredible. And you mentioned only a fraction of myosin heads might be attached at any given moment during contraction.
Yeah, estimates suggest maybe only 20% to 40%, even during maximal force generation. This is likely due to steric hindrance physical constraints, preventing all heads from binding simultaneously, and the kinetics of the cycle itself. Some heads are attaching, some are stroking, some are detaching.
Even so, muscle is pretty efficient, isn't it, converting ATP energy to work?
Remarkably efficient, actually. In isolated muscle preparations under optimal conditions, it can reach efficiencies of around 65%, meaning 65% of the energy from ATP hydrolysis is converted into mechanical work.
And in a living person exercising.
In humans doing steady-state exercise, the measured mechanical efficiencies typically range from 40% to 57%. That's still incredibly good for a biological motor, a real powerhouse of efficiency. This naturally leads us to think about why different muscles behave differently. Why are some built for speed and power, while others are endurance champions?
Right, like eye muscles versus postural muscles.
Exactly. Skeletal muscle fibers aren't all the same. They're broadly classified into 2 main groups based on their speed of contraction: fast twitch and slow twitch fibers.
And the speed difference is significant.
Oh yeah, that lateral rectus muscle moving your eye contracts in maybe 8 milliseconds. The soleus muscle in your lower leg, crucial for posture, takes around 90 milliseconds to reach peak tension. Your calf muscle, the gastrocnemius, has a mix and takes about 40 milliseconds.
What determines that speed difference? Is it the cross-bridge cycle?
It comes down directly to the myosin ATPase activity within those fibers.
The rate at which myosin hydrolyzes ATP.
Exactly. Fast twitch fibers have specific versions, isoforms of the myosin heavy chain protein that chop up ATP incredibly quickly. That allows for rapid cross-bridge cycling and thus fast contraction.
And slow twitch fibers.
They have myosin isoforms that hydrolyze ATP much more slowly. Same basic structure, but slight differences in amino acid sequence lead to slower cycling and slower contraction speed.
So it's baked into the type of myosin they have. What determines which type of myosin a fiber makes?
This is fascinating. It's largely determined by the motor nerve that innervates the muscle fiber.
The nerve tells the muscle what type to be.
To a large extent, yes. It's very difficult to dramatically change a fiber type, say from slow to fast, just through normal exercise.
You can't turn your soleus into an eye muscle by training.
Not really, no.
Yeah.
But classic experiments involving cross-innervation showed this nerve influence powerfully.
What did they do?
They surgically swapped the nerves. They took the nerve that normally goes to a fast muscle and connected it to a slow muscle, and vice versa.
And what happened?
The slow muscle, now getting signals from a fast nerve, actually sped up. Its contraction speed and myosin ATPase activity increased, effectively changing its phenotype towards fast twitch.
Wow. And the fast muscle getting slow signals.
It slowed down. Its shortening velocity and ATPase activity decreased. It vividly demonstrated this profound trophic effect. The nerve's pattern of activity shapes the muscle's characteristics.
Okay, so in humans, what are the main fiber types based on this myosin difference?
We primarily classify them based on their myosin heavy chain, MHC composition. You have type I fibers. Those are your slow-twitch oxidative fibers.
High endurance?
Got it. Type I is slow. Then you have the fast-twitch types. Type 3 are considered fast oxidative glycolytic. They're fast, but also have good endurance.
Type 3 equals fast intermediate endurance.
And type 3 are the really fast, primarily glycolytic fibers, powerful bursts, but fatigue more quickly.
Type Theseus fastest, in humans mostly, low endurance. What about type IB?
Type IB is common in smaller animals like rodents, and it is the fastest isoform known. But it's actually very rarely found in adult human skeletal muscle. Our fastest is generally IOX.
Interesting. Can fibers have a mix?
Yes, you can find hybrid fibers that express more than one type of myosin heavy chain, like type 3 or type 3 NEX. This shows there's some flexibility and transition possible.
And training influences this.
It seems so. Endurance training tends to promote a shift towards more oxidative types, maybe from IX towards IA, or even increasing the proportion of type 1. Strength training tends to favor the maintenance or increase of type 2 fibers.
Okay, so these different fiber types have distinct properties beyond just speed.
Absolutely. They're like specialized tools. Refers to table 12.1 notionally. Type I fibers, being slow, have that slow myosin ATPase. They have moderate SR calcium pumping.
But really high endurance feature.
Exactly. High capillary density for oxygen supply, tons of mitochondria for oxidative metabolism, moderate glycolytic capacity, all geared towards being incredibly resistant to fatigue. Perfect for posture, long-distance running.
And type Pia, the intermediate ones.
They're fast, have high myosin ATPase activity. They also have pretty high capillary density and good mitochondrial content, plus high glycolytic capacity. So they offer balanced speed, power, and decent fatigue resistance. Good for middle-distance events, team sports.
And type like the sprinters.
Even faster ATPase activity.
Right.
But they have lower capillary density, fewer mitochondria, lower oxidative capacity. Their strength is very high glycolytic capacity. Suited for explosive, short burst, sprinting, jumping, heavy lifting. They fatigue relatively quickly.
Makes sense. How are these organized with the nerves? You mentioned motor units.
Right. A motor unit is one alpha motor neuron in the spinal cord and all the muscle fibers it connects to.
And all fibers in one unit are the same type?
Typically, yes. All fibers within a single motor unit usually express the same myosin isoform. So you have slow motor units containing type I fibers and fast motor units containing type CE or ICE fibers.
Are the motor units different sizes?
Yes, significantly. Slow motor units are innervated by smaller motor neurons, which are easier to excite electrically. These units usually contain fewer muscle fibers, maybe 100 to 500.
Okay, smaller, easier to activate.
Fast-twitch motor units are innervated by larger motor neurons that have a higher activation threshold, need a stronger signal to fire. And these units contain many more muscle fibers, maybe 1,000 to 2,000.
Larger, harder to activate, more fibers.
Exactly. And this leads directly to the size principle of recruitment.
Explain that.
When you need to generate force, your nervous system recruits the motor units in order of size. Small, slow motor units are activated first for low-force tasks.
Like standing or walking slowly.
Right. As you need more force, say, to pick up something heavier or start jogging or jump, your brain sends stronger signals, activating those larger motor neurons and bringing the bigger, faster motor units into play progressively.
So you scale the force efficiently by adding larger units as needed.
It's an incredibly efficient system. Slow units for endurance and fine control. Fast units layered on top for power.
This explains why postural muscles are mostly slow twitch.
Exactly. They need that high oxidative capacity, low energy cost, and fatigue resistance for sustained activity. Their smaller fiber diameter and higher capillary density help with oxygen diffusion too.
And muscles for jumping are mostly fast twitch.
Generally, yes. They need rapid, forceful contractions. They typically contain more fibers per unit and have larger diameter fibers, allowing them to generate much greater force.
Are there other differences between fast and slow fibers besides myosin and metabolism?
Oh yeah, lots of subtle but important ones. Even the neuromuscular junction where the nerve meets the muscle is different.
How so?
Fast muscle junctions are built for speed and reliability under high demand. They have more acetylcholine vesicles ready to release, high receptor density, faster acetylcholine breakdown, higher sodium channel density.
Giving them a higher safety factor, meaning they're more likely to fire an action potential.
Initially, yes.
Yeah.
A higher safety factor for reliably triggering that action potential. However, interestingly, that safety factor tends to drop more quickly with repetitive stimulation fast muscle compared to slow muscle.
Huh. What about the SR?
The sarcoplasmic reticulum is much more extensive and developed in fast muscle.
More calcium storage and release machinery.
Yes. Higher levels of RYR1, higher sarcopump density, more stored calcium, even a higher ratio of CAVI 1.1 voltage sensors to RYR1 release channels. All of this contributes to a larger and faster calcium transient, a quicker, bigger spike in calcium concentration.
Which is critical for those fast, forceful contractions.
Absolutely. Drives the faster cross-bridge cycling.
And does calcium sensitivity differ? Does it take less calcium to activate slow fibers?
That's another key difference. Yes. Slow fibers generally begin to develop tension at lower calcium concentrations than fast fibers. They are more sensitive to calcium.
Why is that? Is it troponin C again?
Troponin C plays a role. Remember, the slow isoform of troponin C has only one low-affinity calcium binding site, whereas the fast isoform has two. That difference contributes.
But it's not just troponin C.
No, it's more complex.
Yeah.
Differences in the isoforms of troponin T and tropomyosin also influence calcium sensitivity. It's an integrated effect of the whole thin filament protein complex.
And didn't you mention phosphorylation of the myosin light chain earlier?
Yes, phosphorylation of that regulatory light chain can also increase calcium sensitivity, and this effect seems to be more pronounced in fast muscle fibers. Another layer of regulation. #6. Fine-tuning force and muscle plasticity.
Okay, so putting it all together, how does your brain control how much force your muscle produces? We touched on recruitment.
Recruitment is a major way, yes. Simply activating more motor units. Since all the fibers in a unit contract together, more units means more fibers contracting, means more force.
And following the size principle. Start small and slow, add bigger and faster units as needed.
Exactly. It's like turning up a dimmer switch, but in discrete steps by adding more motor units.
What's the other main way to increase force from fibers that are already active?
That's tetany, which involves increasing the frequency of action potentials firing down the motor nerve to the muscle fiber.
Okay, explain how frequency matters. A single action potential causes just a brief twitch, right?
Right, because the calcium is pumped back into the SR very quickly, so the contraction is short-lived.
But if another action potential arrives before the muscle has fully relaxed from the first one?
Then the mechanical responses, the forces, begin to add together. This is called summation. The second twitch builds on the first.
Okay, and if you send action potentials really fast?
At high stimulation frequencies, the calcium levels inside the muscle fiber don't have time to drop back down between stimuli. They stay elevated.
So the cross bridges keep cycling continuously.
Exactly. And the force developed becomes sustained and much, much greater than the force of a single twitch. This smooth, sustained maximal contraction is what we call fused tetany or tetanus.
Like holding a heavy object steadily.
That involves tetanic contractions in the recruited motor units, yes. If the frequency is slightly lower, so calcium does dip a bit between stimuli, but summation still occurs, you get an unfused or incomplete tetany where the force might ripple slightly.
So higher frequency leads to smoother, stronger contractions up to a maximum.
Correct. It's the fusion of those individual twitches due to sustained high calcium.
But here's something interesting. Why is a single twitch so much weaker than a tetanic contraction? You'd think if enough calcium is released even in a twitch, it should allow maximum cross-bridge formation, shouldn't it?
That's a great question. And the answer likely lies in the series elastic component of the muscle.
The stretchy parts, like tendons and connective tissue.
Yes, but it also includes elasticity within the muscle fiber itself. Things like the titin protein and even the myosin cross bridges have some elasticity.
Okay, so how does elasticity explain the difference between twitch and tetany?
During a very brief twitch, the contractile elements, the actin and myosin sliding, might generate force quickly, but they don't have enough time to fully stretch out all those series elastic components before the calcium drops and relaxation begins.
So some of the force is absorbed internally by stretching these elements, and less is measured externally.
Exactly. The externally measured tension is submaximal because the internal stretching isn't complete. With tetany, however, the calcium stays high for longer.
Giving the contractile elements enough time to fully stretch those elastic components.
Precisely, allowing the full force generated by the cross bridges to be transmitted and measured externally. So tetanic force represents the maximal force generating capacity once elasticity is accounted for.
That makes sense. Does the frequency needed for tetany differ between fiber types?
Yes, it does. Slow fibers, because their twitches are inherently longer duration.
They take longer to contract and relax.
Right. They can achieve fused tetany at lower stimulation frequencies compared to fast fibers, which need much higher frequencies due to their quicker twitch dynamics.
And we know fast fibers generally produce more force overall.
Fast motor units do, yes, partly because they have larger diameter fibers and partly because each fast motor unit contains more fibers. But even if you compare force per unit of cross-sectional area, type 2 fibers still produce about 20% more isometric tension than type I fibers.
So type 2 units really are specialized for those high-force bursts.
It's well suited for it.
Now, beyond conscious control of recruitment and frequency, our body has built-in reflexes too, right? To help modulate force and stability.
Absolutely. Very sophisticated feedback loops involving sensory receptors within the muscles and tendons.
Like muscle spindles.
Muscle spicules are key. They're specialized sensory fibers, also called intrafusal fibers, that lie parallel to the main extrafusal muscle fibers.
And they sense stretch.
Exactly. They detect changes in muscle length and the speed of that change. When a muscle is stretched, the spindles are also stretched, and they send signals back to the spinal cord via sensory nerves.
What does the spinal cord do with that signal?
It directly excites the alpha motor neurons that go back to the same muscle that was stretched.
Causing a reflex contraction? The stretch reflex?
That's the one. The classic knee-jerk reflex is an example. It helps automatically resist unexpected stretches, maintain posture, and provides constant feedback to the brain about muscle length.
And the spindle adjusts itself too, right? So it works at different muscle lengths.
Yes, it receives its own motor input, gamma motor neurons, that takes up the slack as the main muscle shortens, keeping the spindle sensitive to stretch across the full range of motion.
Clever. What's the other main sensory receptor?
The Golgi tendon organs, or GTOs.
Located in the tendons, as the name suggests.
Exactly, right where the muscle fibers connect to the tendon. They are arranged in series with the muscle fibers, not parallel like spindles.
So they sense tension or force?
Precisely. They respond to the tension generated when muscle fibers contract and pull on the tendon. They are sensitive to the contractions of even individual fibers connected to them.
And they send signals back to the spinal cord too, via type afferent nerves.
Yes. But unlike the spindle reflex, the GTO input typically causes inhibition of the alpha motor neurons going to the contracting muscle.
Inhibition. So it reduces the drive to contract.
Generally, yes, via inhibitory interneurons in the spinal cord. It also tends to excite the motor neurons of the antagonistic muscle.
Why would it do that? Seems counterintuitive.
It's thought to act like a protective mechanism, preventing excessive force generation that might damage the tendon or muscle. It might also help to smooth out muscle contractions by modulating motor unit recruitment, preventing jerky movements.
But doesn't that inhibition limit maximal force?
Interestingly, the GTO response isn't linear. Its firing rate tends to plateau or even decrease at very high force levels. So its inhibitory influence might actually lessen during maximal efforts, potentially allowing for greater motor unit recruitment when you really need it.
So it's more complex than just a simple break.
Much more complex, likely involved in fine motor control and adapting its role depending on the context of the movement.
Okay, what about muscle tone? That feeling of slight firmness in muscles even at rest. Where does that come from?
That resting muscle tone is largely due to low levels of ongoing contractile activity in a small number of motor units.
Driven by reflexes.
Primarily driven by the tonic activity of those muscle spindles we discussed. That constant sensory feedback leads to a baseline level of motor neuron firing and thus slight muscle tension.
So if you cut the sensory nerves from the spindles,
the muscle tone disappears, the muscle becomes completely flaccid, shows how important that reflex arc is for maintaining readiness. Which brings us to a critical question. How do muscles actually fuel all this incredible activity? Contraction needs energy.
ATP, right. Adenosine triphosphate. That's a direct fuel source.
Absolutely. Every cycle of the cross bridge, every pump action like Circa, uses ATP. But here's the catch. The amount of ATP readily available, just sitting there in the muscle cell, is incredibly small.
Only enough for a few seconds of intense activity.
Maybe even less, just a few contractions if it weren't constantly replenished. But the amazing thing is, it is constantly and very efficiently refilled during contraction.
So ATP levels don't actually drop that much, even during hard work.
Surprisingly, no. Even when a muscle is significantly fatigued, the overall ATP concentration is usually only modestly decreased from resting levels. The replenishment mechanisms are that good.
Okay, so what's the first line of replenishment? The quickest way to make more ATP.
That's creatine phosphate, also called phosphocreatine.
Ah, the stuff athletes sometimes supplement with.
Exactly. Muscle cells store a reserve of this high-energy phosphate compound. There's an enzyme, creatine phosphokinase, CPK, that can rapidly transfer the phosphate group from creatine phosphate directly onto ADP, regenerating ATP almost instantly.
Like a quick charge battery right inside the cell.
That's a perfect analogy. It provides immediate ATP resynthesis, especially crucial for the very start of exercise or intense bursts. Some CPKs even located right near the myosin heads for ultra-fast refueling where it's needed most.
How long can the system power maximal activity?
Not very long. The creatine phosphate store, while larger than the ATP store, is still limited maybe 5 times the ATP pool. It can only support maximal effort for maybe less than a minute. Some say only 10-15 seconds.
So fatigue during really intense sprints or lifts might be linked to running low on creatine phosphate.
Its depletion is definitely correlated with fatigue in that type of short maximal exertion.
Okay, so after creatine phosphate runs low, what's next for fueling longer activity?
For more sustained work, muscles rely heavily on breaking down carbohydrates and fatty acids.
Started with carbs. Muscles store glycogen, right?
Yes. Muscle cells store a significant amount of glycogen, which is just branched chains of glucose molecules. This glycogen can be broken down into glucose units.
And that glucose can be used in 2 ways.
Right. It can go to oxidative phosphorylation within the mitochondria. This requires oxygen and is very efficient, yielding about 36 or 37 ATP molecules per glucose molecule.
The aerobic pathway.
The aerobic pathway, yes. Or, if oxygen is limited, or energy demand is extremely high, glucose can be partially broken down via glycolysis in the cytosol.
The anaerobic pathway.
Correct. Glycolysis doesn't require oxygen, but it's much less efficient, yielding only 2 or 3 ATP per glucose, plus lactate as a byproduct.
Muscles can also grab glucose from the blood.
Yes, they can take up glucose circulating in the blood, and this process is stimulated by the hormone insulin and also by exercise itself.
And fatigue during prolonged events, like a marathon, is often linked to running out of glycogen.
Yes, hitting the wall in endurance events is often associated with the depletion of these crucial muscle glycogen stores. The body then has to rely more heavily on other fuels, which can't necessarily supply energy quite as rapidly.
Which brings us to fats. When do they become important?
Fatty acids become a major energy source during lower intensity, longer duration exercise, and also when glycogen stores start to run low.
Where do the fatty acids come from?
Muscles store some fatty acids and also store triglycerides internally, which can be broken down, hydrolyzed, to release fatty acids. They can also take up fatty acids circulating in the blood, which come from the breakdown of fat stored elsewhere in the body.
And how are fatty acids used for energy?
They are transported into the mitochondria, a process involving carnitine, and then broken down through a process called beta oxidation. This feeds into the same oxidative phosphorylation pathway as glucose breakdown products.
Is using fat efficient?
It yields a huge amount of ATP per molecule, so it's very energy dense. But it's strictly an aerobic process. It requires oxygen, and the rate of ATP production from fat is generally slower than from carbohydrate.
So good for long, slow efforts, less so for sprints.
Generally, yes. There's always a mix of fuels being used, but the relative contribution shifts depending on intensity and duration.
Okay, what happens when you're working so hard that your oxygen supply can't keep up with the demand? You mentioned anaerobic glycolysis.
Right. When energy demands exceed the capacity of aerobic metabolism, you start relying more on anaerobic glycolysis producing lactate. This leads to what's called an oxygen debt.
Oxygen debt, meaning you have to pay back the oxygen later.
Exactly. After you stop exercising, your breathing rate and oxygen consumption remain elevated above resting levels for some time. This is the recovery oxygen, or excess post-exercise oxygen consumption, EPOC.
What's that extra oxygen used for?
It's used to repay the debt incurred during exercise. It helps restore metabolite levels, replenishing creatine phosphate stores, rebuilding ATP, converting lactate back to glucose or glycogen, some in the liver, some in muscle, and restoring oxygen levels in blood and myoglobin. Plus, body temperature is elevated, which increases metabolic rate.
And this oxygen debt is bigger after harder exercise.
Much bigger, yes. Strenuous exercise relies more heavily on those fast glycolytic motor units and anaerobic pathways, so a larger debt is incurred. It's essentially a measure of the energy that was supplied anaerobically during the workout.
Okay, let's talk fatigue. We use the term all the time. But what actually causes it at the muscle level? You said it's not ATP depletion?
Right, that's a critical point often misunderstood. While energy supply is related, fatigue isn't typically caused by running out of ATP. ATP levels are actively protected.
So fatigue is more like a protective mechanism.
That's a prominent view, yes. It's seen as a complex physiological response designed to prevent ATP from dropping to dangerously low levels that could actually damage or kill the muscle cell. It forces you to slow down or stop before that happens. Muscles seem to have evolved redundant systems to ensure ATP homeostasis is maintained.
So if not lack of ATP, what does contribute to that decline in force during fatigue?
Many factors are implicated, and it depends on the type of exercise. During brief, really intense contractions like maximal tetany,
Especially in fast motor units?
yes, the decline in force is rapid and almost complete. This correlates well with the rapid depletion of creatine phosphate and glycogen stores, and importantly, the accumulation of metabolic byproducts.
Like lactic acid, does that directly cause fatigue?
Lactic acid itself quickly dissociates into lactate and hydrogen ions, H+. It's the accumulation of H+ caret leading to a decrease in muscle pH acidosis that's strongly implicated.
How does acidity affect contraction?
It seems to inhibit actin-myosin interaction in several ways. It might alter calcium binding to troponin C, making it less sensitive, and it might directly reduce the maximum force or number of strongly bound cross bridges.
What else accumulates?
Inorganic phosphate, pi, from the breakdown of ATP and creatine phosphate, also accumulates significantly. High pi levels are another major suspect in causing fatigue.
How does phosphate interfere?
It's thought to directly decrease the force produced by each individual cross bridge, potentially reduce calcium release from the SR, and might also interfere with actin-myosin binding kinetics.
So it's a combination of acidity and phosphate buildup.
Those are considered major players in high-intensity fatigue. Other factors likely contribute too, like localized ADP increases near the contractile proteins, potassium ions building up outside the cell membrane, which can affect excitability, and maybe even reactive oxygen species generated during metabolism.
What about fatigue during long endurance exercise? Is it the same factors?
Less likely to be acidosis or phosphate accumulation in the same way. There, depletion of glycogen stores seems more critical. Also, factors related to calcium handling might become important, perhaps reduced SR calcium release or uptake over long periods. And central nervous system fatigue definitely plays a larger role in endurance performance.
Central fatigue, meaning the brain itself gets tired.
Not necessarily tired in the muscle sense, but changes in brain neurotransmitters or signaling pathways can reduce the drive from the central nervous system to the muscles, contributing to the perception of fatigue and limiting performance.
So putting this into perspective for everyday life or athletes, when we feel tired during exercise, is it usually our muscles truly fatiguing at the cellular level?
For most people, probably not completely. What we often perceive as general physical fatigue is more likely related to those broader homeostatic disturbances, maybe falling blood glucose, accumulation of metabolites circulating in the blood, perceived exertion, psychological factors. The central nervous system plays a huge role in this overall feeling.
But highly trained athletes can push closer to actual muscle fatigue.
Yes, highly motivated individuals can often override those initial feelings of discomfort and push their muscles much closer to their physiological limits, where cellular fatigue mechanisms do become significant. Part of elite performance is actually training the brain and boosting those motivational factors to tolerate higher levels of discomfort and push closer to true physiological limits. #8, growth, development, and repair of skeletal muscle.
Okay, let's shift gears a bit. How do muscles actually grow and develop, and how do they adapt to things like exercise or disuse? And what happens when they get injured?
Right, the plasticity of muscle. Where does it start? Do fibers differentiate early? They do. Muscle fibers actually start to differentiate and take on some characteristics even before they get connected to a nerve. Initially, they tend to resemble slow type I cells physiologically. Their membrane has acetylcholine receptors spread pretty widely over the surface.
But then the nerve arrives.
When the first nerve terminal makes contact, a specialized region called the end plate or neuromuscular junction forms, and those acetylcholine receptors cluster dramatically right there, under the nerve terminal.
And you said the nerve then determines the fiber's fate.
Largely, yes. Small tonically active motor neurons tend to promote the development of slow, type I oxidative motor units. Larger motor neurons that fire in faster bursts lead to the formation of fast type II motor units.
So the nerve's activity pattern sculpts the muscle fiber type.
It drives major cellular changes, including switching on the genes for the specific fast or slow myosin isoforms, replacing the embryonic or neonatal versions that were there initially. It's that trophic effect again.
And muscles obviously grow as we grow. How does that happen? Do we get more fibers?
Generally, no. The number of muscle fibers is thought to be largely set shortly after birth. Growth in muscle length, as our skeleton grows, happens mainly by adding new sarcomeres in series at the ends of the existing muscle fibers.
Like adding more links to a chain.
Exactly. And this process is reversible. If you immobilize a muscle in a shortened position, it can actually remove sarcomeres from the ends to adapt its length.
Does changing length affect force?
It affects the muscle's optimal length for tension and its maximum shortening velocity, but not necessarily the maximum force it can generate per unit area.
So how does muscle get stronger and thicker during growth or with training?
That's primarily due to hypertrophy.
Meaning the existing fibers get bigger?
Precisely.
Yeah.
Hypertrophy involves adding more myofibrils and thus more sarcomeres in parallel within each muscle fiber. This increases the fiber's cross-sectional area and diameter.
And that increases the force it can generate.
Yes, because force is proportional to the number of cross bridges acting in parallel. Doubling the myofibrillar diameter can potentially double the force, but it doesn't really affect the maximum shortening velocity, V0.
And resistance exercise is the big stimulus for hypertrophy.
It's a major promoter, yes. Lifting weights, resistance training, that mechanical stress triggers hypertrophy. Understanding the signaling pathways here is key for, mastering how muscles adapt. Hypertrophy happens when protein synthesis rates exceed protein degradation rates.
What signals turn on protein synthesis?
During development, and especially in response to resistance exercise, a key player is insulin-like growth factor 1, or IGF-1. Elevated IGF-1 levels strongly stimulate hypertrophy, primarily through a signaling cascade called the ACT-mTOR pathway.
ACT-mTOR. That's a central growth pathway.
A major one in many tissues, yes. But mechanical force itself, the tension from contraction, is also a powerful stimulus.
How is force sensed?
It's thought to be transduced or converted into biochemical signals through those adhesion complexes we talked about, like the dystrophin glycoprotein complex, which link the internal cytoskeleton to the outside world. They sense the strain.
And other molecules are involved too, like microRNAs.
Yes. The regulation is complex. Tiny RNA molecules like microRNAs and also long non-coding RNAs produced during exercise seem to influence these growth pathways as well. It's an active area of research.
What about endurance exercise? Does that cause hypertrophy?
It can cause some, yes, but its primary effects are different. Endurance training mainly promotes increased oxidative capacity.
More mitochondria, better oxygen use.
Exactly. And improved blood flow through angiogenesis, the growth of new capillaries supplying the muscle fibers.
What signals drive those changes?
A key molecule here seems to be a transcription coactivator called PGC1I. It gets activated by endurance exercise and turns on genes involved in mitochondrial biogenesis and oxidative metabolism. Angiogenesis seems to involve factors like VEGF, which is downstream from PGC1.
But you said standard exercise doesn't really convert fiber types, like type 2 to type 1.
Generally not a complete conversion, no. You might see shifts within the type 2 subtypes, like more II and less MERX with endurance training, or vice versa with D-training or strength focus. You see those hybrid fibers. But turning a fully differentiated type 2 fiber into a type 3 requires more extreme interventions, like chronic low-frequency electrical stimulation mimicking a slow nerve, or those cross-innervation surgeries. Normal exercise doesn't typically provide that kind of stimulus. On the flip side, atrophy is when muscle wastes away.
When protein degradation wins out over synthesis.
Exactly. This happens rapidly during periods of disuse, like prolonged bed rest, limb immobilization in a cast, or the microgravity of spaceflight. A muscle mass and strength decrease quickly.
What pathways drive atrophy?
Disuse atrophy involves both shutting down protein synthesis pathways, like ACT-mTOR, and ramping up protein degradation pathways. A major degradation pathway involves the ubiquitin proteasome system, regulated partly by the FOXO family of transcription factors, which turn on atrogene's genes promoting atrophy. Increased activity of proteases like caspase-3 is also involved.
Does atrophy affect fiber types differently?
Interestingly, yes. During unloading or disuse, slow motor units, type I fibers, tend to atrophy more rapidly and significantly than fast motor units.
Really? Why would that be?
It's thought that slow fibers rely more on continuous neural activity and weight-bearing load to maintain their size and phenotype. When that's removed, they suffer more.
And does this affect their properties?
Yes. Besides the obvious decrease in maximal force due to shrinking, these atrophying slow fibers paradoxically often show an increase in their maximal shortening velocity.
They get faster as they shrink.
It seems counterintuitive, but yes. It's because they start expressing faster myosin isoforms, shifting their phenotype slightly towards a faster type even as they lose mass.
Which is a big issue for astronauts, right?
A critical issue. Designing effective exercise countermeasures to prevent this atrophy and phenotypic shift during long space flights is a major challenge in space medicine.
Okay, what about muscle repair after an actual injury, like a tear strain?
Muscle has a remarkable capacity for repair. For minor injuries, maybe small tears in the sarcolemma that cause a little calcium leak,
Yeah.
those can often be rapidly patched up by proteins like dysferlin working together with annexin 2 to sort of form a membrane patch.
But for bigger injuries, more extensive damage.
That triggers a more elaborate regeneration process.
Yeah.
And the absolute stars of this show are the satellite cells.
Muscle stem cells.
Exactly. They normally sit quietly under the basal lamina, the outer sheath surrounding the muscle fiber. But when the fiber is damaged, signals are released that activate these satellite cells.
What do they do once activated?
They wake up, start dividing rapidly, proliferate. Then they begin to mature and differentiate into committed muscle precursor cells,
Yeah.
myoblasts. Then these myoblasts fuse together to form new multinucleated structures called myotubes. Initially, these myotubes have their nuclei clustered in the center.
Like immature muscle fibers.
Precisely. These myotubes then continue to mature, grow, start producing contractile proteins, and eventually become functional skeletal muscle fibers. As they mature, the nuclei migrate outwards to their normal position just under the sarcolemma.
And do these regenerated fibers just replace the damaged ones?
They can fuse with existing damaged fibers to repair them, or fuse together to form completely new fibers if the damage was severe enough to cause fiber death.
Does the immune system get involved?
Critically involved.
Yeah.
It's an orchestrated response. Early on, M1 macrophages come in to clear away the cellular debris from the injury site, and they also release factors that stimulate satellite cell proliferation.
So they clean up and kickstart the repair.
Right. Then later, a different type, M2 macrophages, become more prominent. They promote the differentiation and fusion of the myoblasts into myotubes, helping with the rebuilding phase.
Fascinating coordination. How can you tell if a fiber has regenerated?
Regenerated fibers often have those centrally located nuclei for a while, which is unusual in mature fibers. They also temporarily express embryonic or neonatal myosin isoforms before switching to the mature type determined by the nerve supply.
So they'll eventually become type I or type 2 based on the nerve that reinnervates them.
Exactly. The nerve dictates their final phenotype once regeneration is complete.
Okay, one last thing in this section. That muscle soreness we get after a hard workout. DOMS.
Yes. Delayed onset muscle soreness. Familiar to many.
What exactly causes that dull, aching pain that peaks like a day or two after the exercise?
It's particularly common after eccentric exercise, where your contracting muscles are actually lengthening under load.
Like running downhill or lowering a heavy weight slowly.
Exactly. Those vigorous stretching contractions seem to cause microscopic damage or injury to the muscle fibers themselves, and possibly the connective tissue.
So the soreness is from actual injury.
Yes, it's associated with inflammation, swelling, and damage within the muscle cells, often observed near the myotendinous junction. It's not just lactic acid buildup, which clears relatively quickly. This is actual structural disruption.
Does it affect certain fiber types more?
It seems type 2 motor units are often affected more severely,
Why them?
probably because they generate higher maximal forces. During eccentric contractions, the load imposed can actually exceed the maximal force the muscle can actively generate, putting a huge strain on those high-force type 2 fibers, making them more susceptible to damage.
And recovery takes time.
Yes. DOMS is associated with reduced range of motion, stiffness, and temporary weakness. Recovery is relatively slow because it involves the repair and regeneration of those injured sarcomeres and cellular structures. #9. Biophysical properties of skeletal muscle. To really understand muscle function at a quantitative level like scientists do, we need to touch on some key biophysical concepts. Two fundamental types of contractions are studied, isometric and isotonic.
Okay, isometric means.
Same length. In an isometric contraction, the overall muscle length is held constant, for example, by fixing both ends. What's measured is the force or tension the muscle generates at that fixed length.
Like pushing against that immovable wall. I generate force, but the muscle doesn't shorten.
Exactly. Now, isotonic means same tone or same force.
So the force is constant?
Yes. In an isotonic contraction, the muscle shortens or lengthens against a constant load or force. What's measured here is the change in muscle length or the velocity of that change.
Like lifting a dumbbell at a steady pace. The load is constant, and you measure how fast the muscle shortens.
Precisely. These two types of measurements reveal fundamental properties of muscle.
Like the length-tension relationship.
That's a classic one, usually studied under isometric conditions. If you take a muscle at rest and just passively stretch it, it resists that stretch.
Because of its elasticity, the connective tissue and tighten.
Right. That generates a passive tension curve that rises steeply as you stretch it further. But the interesting part is the active tension.
The force generated when you stimulate it to contract at different lengths.
Exactly. When you stimulate the muscle isometrically at different starting lengths, you find that the active contractile force it generates changes dramatically with length.
How does it change?
Force increases as you stretch the muscle from a short length up to a certain point.
The optimal length, LO?
Right, LO. At this optimal length, the muscle generates its maximum active tension. But if you stretch it beyond LO, the active force actually starts to decrease again.
So there's a sweet spot for force generation. Why? What does this tell us?
This provides incredibly strong support for the sliding filament theory. It relates directly to the overlap between actin and myosin filaments within the sarcomere.
Ah, so at very short lengths.
If the sarcomere is too short, below about 2 micrometers, the thin filaments from opposite ends might collide or crumple against the Z-lines, interfering with optimal cross-bridge formation. Force drops off.
And at very long lengths, beyond LO, like 3.7 micrometers.
The actin and myosin filaments are pulled so far apart that there's very little overlap between them. Fewer myosin heads can reach the actin binding sites, so fewer cross-bridges can form. Again, force drops off.
So LO represents the length where the actin-myosin overlap is maximal, allowing the maximum number of cross-bridges to form and generate force.
Exactly. It's a beautiful demonstration of how the microscopic structure directly dictates the macroscopic mechanical properties.
Okay, what about the force-velocity relationship? That's usually studied under isotonic conditions.
Correct. This looks at how the speed or velocity at which a muscle shortens depends on the load it's trying to move.
And intuitively, you'd think heavier loads means slower shortening.
Precisely. If there's no load at all, zero force, the muscle shortens at its absolute maximal velocity. We call this V0 or Vmax.
And V0 depends on the fiber type.
Yes, V0 is directly proportional to the maximal rate of cross-bridge cycling, which in turn depends on that myosin AT pace activity. So fast-twitch fibers have a much higher V0 than slow-twitch fibers.
Okay, so maximal speed with no load. What happens as you add load?
As you progressively increase the load, the force the muscle has to overcome, the shortening velocity steadily decreases.
Makes sense.
Eventually, you reach a load that the muscle can just barely lift but cannot shorten against anymore. At this point, the shortening velocity drops to 0.
And that force where velocity is 0 is the maximal isometric tension.
Exactly. PO0, the maximal isometric tension. This is proportional to the total number of active cross-bridges attached to that moment. And as we noted, this is generally higher for fast motor units due to fiber size and number.
So faster fibers aren't necessarily stronger in terms of isometric force per area, but they are much faster, especially at low loads.
Correct. Now, what about muscle power? Power is force times velocity.
Where is power output maximal? Is it at 0 load, max velocity, or max load, 0 velocity?
Neither. Power is 0 at both extremes. Maximal power output is actually achieved at an intermediate load, typically around 30% of the maximal isometric tension.
So lifting a moderate weight relatively quickly generates the most power.
Exactly. That's the sweet spot for maximizing the rate of work done by the muscle. If you continue to increase the load beyond Piedro, the muscle can actually be forcibly lengthened while still trying to contract that's eccentric contraction, demonstrating a negative velocity. #tag outro.
So wrapping this all up, what does this deep dive really mean for understanding our muscles? We've covered a huge amount of ground.
You really have.
We journeyed from the big picture, the gross anatomy of muscle attaching to bone,
Right down through that intricate hierarchy, fascicles, fibers, myofibrils.
down to the molecular dance of actin and myosin within the sarcomere itself, the absolute core of contraction.
And we unpack that crucial role of calcium, the whole complex ballet of excitation-contraction coupling.
Including the vital importance of proteins like dystrophin, highlighted by conditions like DMD, the structural integrity piece.
Absolutely. Then we explored the fascinating diversity, the differences between fast and slow muscle fiber types, and how they suit different tasks.
And how your body masterfully modulates force, using recruitment based on the size principle, and by adjusting firing frequency to achieve tetany.
Plus those incredible internal reflex systems, the spindles and Golgi tendon organs, constantly providing feedback and stability without us even thinking about it.
Then we delved into how muscles are fueled from creatine phosphate bursts to sustain aerobic metabolism of carbs and fats.
And the mechanisms of fatigue, crucially, understanding it less as running out of fuel and more as a complex protective mechanism preventing cellular damage.
Right, that was a key insight. And finally, the remarkable plasticity of muscle, how it grows through hypertrophy, develops specific characteristics based on nerve input, adapts to training.
And even repairs itself after injury, using those amazing satellite cells. We saw how both resistance and endurance training drive specific adaptations via distinct signaling pathways.
This level of detail going through each step really provides that expert-level insight we were aiming for.
Hopefully, yes.
Yeah.
Because that knowledge, really understanding the why behind the what, is where the true value lies, especially if you want to apply it.
It's just undeniable that skeletal muscle is an absolute masterpiece of biological engineering, capable of incredible feats, constantly adapting.
It truly is.
Understanding these fundamental principles just gives you a whole new appreciation for every single movement you make, doesn't it? From the tiniest finger twitch to lifting something heavy.
It certainly does. And maybe that leaves us with a final thought, a question for you, our listeners, to ponder.
Go for it.
How might that inform real future advancements? Think about areas like designing truly personalized exercise programs tailored to individual physiology, or developing much more effective rehabilitation strategies after injury, or even creating breakthrough treatments for devastating conditions like the muscular dystrophies. Where could this fundamental knowledge take us?
That's a fantastic thought to leave everyone with. Where can this deep understanding lead?
Excellent. Well, that's all the time we have for this deep dive into skeletal muscle physiology. We sincerely hope you feel truly well-informed, maybe even closer to that mastery level, and ready to apply this knowledge.
Will be as insightful.
We'll catch you on the next deep dive.